Okay, so last lecture we were talking about stem cells. We talked about embryonic stem cells. And towards the end of the lecture we talked a little bit about adult stem cells. And one thing that I neglected to mention in the lecture on Monday is that we talked about adult stem cells, and we gave an example of the intestinal epithelial stem cells, and that the stem cells reside at the bottom in the crypt of the villi. So these long projections are villi, and at the bottom, basically where this part, where my fingers are villi, is where you find this pocket of stem cells. And so they can undergo, we talked about symmetrical versus asymmetrical. So if they're going symmetrical division, quite frequently they'll divide to give rise to two identical stem cells. That's fine, it can maintain that stem cell population. Or they may receive cues from adjacent cells. This is the part that I didn't mention last lecture, it's important, is that these adjacent cells, and it's not important what their names are, but those cells send signals to the stem cells to decide whether to self-renew or asymmetrically divide to give rise to progenitor cells that can go on and differentiate into mature intestinal epithelial cells. So that's what we refer to as the stem cell niche. So it's the area within the body where the cells reside in the tissue, and it's also cells that are surrounding the stem cells that can influence how the decisions that the stem cells make, whether they want to self-renew or differentiate. So, I briefly talked about how the theory is that stem cells are generally, for example, if we're looking at adult stem cells, they would represent a very small fraction of cells in their body. There's not a lot of them. And as I talked about, they can make a decision to self-renew and give rise to two identical stem cells, or to commit to becoming a progenitor or a differentiated cell. So, and usually as those progenitor cells or differentiated cells in some cases will proliferate. So you will expand the number of cells as the cells move towards their final destination. So small number of stem cells, larger numbers of differentiated cells, and the idea is that it's a one-way ticket down the mountain. Once the cells make it down to the final differentiation stage, they're done. They can't do anything else other than function in the capacity of what that cell type does relevant to the tissue that it resides in. But it turned out that this isn't necessarily always the case, that we can artificially introduce transcription factors. And these transcription factors are called Yamanaka reprogramming factors. They're named after this fellow Yamanaka, who discovered that when you introduce these transcription factors, in this case OCT4, SOX2, ALF4, or and c-Myc, those transcription factors are generally expressed at very high levels in embryonic stem cells. They're not normally expressed to any appreciable level in differentiated cells. But if you were to take differentiated cells, like, for example, skin cells, and you would use viral vectors, in the early studies you use retroviruses, now they use other types that are not quite as potentially harmful, and you express high levels of these four transcription factors, the Yamanaka factors, you can induce these differentiated cells to revert back to a pluripotent state. So remember, pluripotency refers to the ability of a cell to differentiate into all three germ layers. In essence, can form any cell type within the body. So this process is called induced pluripotency, or these cells are referred to as induced pluripotent stem cells, or iPS cells. And so this gathered quite a bit of enthusiasm from the perspective that they have the potential for what we call regenerative medicine. So the ability to replace damaged, lost, or diseased cells. So how would that work? So if we were, for example, to take a patient, and it could be any patient, in this case, the textbook's using an example, maybe somebody who has a brain disorder or a degenerative disorder of the brain where they've lost brain cells, like Parkinson's disease, for example. And you could take skin fibroblasts from this patient, and using viral vectors or other genetic techniques, you could artificially express high levels of the Yamanaka factors in these cultured skin cells. So these are primary cells. You artificially introduce these transcription factors, and what happens is some of those cells will now actually start to revert to a pluripotent state. They form induce pluripotent stem cells. And so you can take these cells and you can do a couple of things. So if you're interested in sort of therapeutic purposes, you could use them to, or differentiate them into neurons, and then use those neurons to introduce back into the patient. But you could do some other clever things. So let's say they have a genetic mutation that even if you take these cells and you put them back into neurons, they still have the disease gene within them. You can use genomic editing, and you can actually repair the mutation. And then now those repaired, genetically corrected cells can be differentiated into neurons and then transplanted back into the patient to replenish the lost cells in the brain. And the advantage of this is that because the cells come from the patient's own body, your body doesn't see it as foreign. It won't reject it. Whereas if you were to take, you could take a healthy person and a different person and do the same thing, but those cells have a very high likelihood, because they come from a different person, to be rejected, as they're perceived as foreign. So this also alleviates some of the ethical problems that are associated with embryonic stem cells. So embryonic stem cells are also pluripotent, and certainly in many animal studies you can show that you can use these embryonic stem cells, differentiate them to certain types of cells, and transplant them into a mouse where maybe there's some artificially induced damage. And that's fine, but the problem is there's ethical considerations if you want to do it in humans, because you essentially have to create a life, sperm and egg, from a zygote and embryo, and then you destroy that embryo to generate the embryonic stem cells. And a lot of people get a little upset about that for ethical reasons. So there's no problems ethically doing this, because you can take patient cells from the own patient, and of course with their consent and permission, we talked about that last lecture, they have to agree to undergo this procedure. Now the other thing that you can do is, some cell types are very difficult to grow in culture. So neurons, for example. You can't, and again, if we wanted to look at human neurons and we want to get a sample, well, it's kind of hard. I don't think too many people want to consent to have a hole drilled in their head and rip some brain tissue out. So one way to work around this is you could take these iPS cells, differentiate them into neurons, and then you can study those neurons in culture and understand what's gone wrong with them. So you could start to understand some of the molecular processes that are messed up from that genetic mutation. And then you could also use them for screening drugs that could potentially alleviate some of the problems, the molecular problems, biochemical problems associated with that mutation. So now you have a much better cell culture model, because it's human cells. They have the mutation. They have some kind of phenotype. Usually in culture, they're very sensitive to certain stressors. And then you could devise a drug screening process to identify drugs that can counter their sensitivity to certain stresses and rescue it and allow those neurons to be healthy. So it also has the great potential as a drug screening type of technology. So we talked a little bit about symmetric versus asymmetric division. What are the types of cells that undergo this? So if we're looking at primary or transformed cells, so primary cells could be a skin fibroblast grown in a dish. Transformed cells could be a cell line coming from a cancer tumor, like HeLa cells, or many, many thousands of other types of cell lines have been established over the last 70 years. And they, for the most part, these cells are, with some exceptions, are in essence already in a differentiated type of state. In terms of their therapeutic potential, with primary cells, it's fairly limited. I mean, it's, in essence, you kind of do that when you're doing tissue transplantation, but in terms of culturing cells and then reintroducing it back into the body, it doesn't work particularly well. There are some exceptions. And of course, with transformed cells, we certainly don't want to be introducing those back into our body because they're cancerous, and we don't want to give ourselves cancer. In terms of symmetric division, we talked quite a bit about stem cells. All stem cells, to various extent, can undergo either symmetric or asymmetric division. Embryonic stem cells and induced pluripotent stem cells, of course, are pluripotent, can give rise to many different types of cells. And as I mentioned, in terms of therapeutic potential, there's a lot of ethical restrictions. For the most part, most regulatory agents will not allow embryonic stem cells to be used for therapeutic purposes. induce pluripotent stem cells. So that, again, have great potential in terms of regenerative medicine. There are some concerns, though, safety concerns. One of the things I forgot to mention is when we look at these transcription factors, one of them, c-Myc, is expressed at very high levels in a lot of different cancer cells. So one of the concerns is when you generate these iPS cells and then you differentiate them, sometimes some cells don't fully differentiate and they retain some of that pluripotency. And they may also retain high-level expression of some of those YAMA-NACA factors. One of the big concerns is c-Myc. And so if those cells incur additional mutation, they may become now much more cancerous and much more... so in essence, these cells, if you use them, do have the potential to become cancerous themselves. So that is one of the biggest concerns with using induced pluripotent stem cells. Adult stem cells, again, we talked about intestinal epithelial adult stem cells. They are not pluripotent, they're what we refer to as multipotent. What is that? It means they cannot give rise to all 3 germ layers. They may only they could give rise to 2 germ layer cell types, or maybe only 1. So they have much more restricted ability to differentiate into a lot of different cell types. In terms of therapeutic potential, absolutely. The initial stem cells, in terms of understanding stem cell biology, 2 Canadian scientists, Till and McCulloch, were the first ones to identify bone marrow stem cells. And through their work back in the 60s and 70s, actually laid the foundation for isolation, propagation of stem cells, and they're routinely used for therapeutic purposes. So in many cases, people that have cancers, or particularly leukemia, they have to do whole body irradiation or very potent chemotherapeutic agent exposure, which generally wipes out their bone marrow. So frequently what they do is before they undergo treatment, is they isolate those stem cells, those bone marrow stem cells, and then propagate them in a dish. They aggressively treat the cancer, and then replenish the bone marrow by introducing those adult stem cells, bone marrow cells, back into the bone marrow, which can reconstitute the whole blood system because they're multipotent stem cells. Okay, so that finishes that aspect of stem cells. We're going to move on now to talk about mitochondria. And just give me a second. Okay, so we're going to talk a little bit about the structure and function of mitochondria. You probably know a little bit about this already. Most of us are quite familiar about metabolism and ATP production by mitochondria. So it's going to be a little bit of a recap. I'm going to be honest, a little bit boring, but we're going to get into some interesting aspects many of you may not appreciate or know about mitochondria. They do way more than make ATP. And we'll talk about some interesting aspects of where do mitochondria come from. Mitochondrial DNA, kind of weird and eclectic, does a lot of cool things. We'll talk about mitochondria. I always think of them as this little perfect round oval. That's really not the case, because they can undergo fusion, they can glom together, and they can break apart. And then we'll talk a little bit at the end about how calcium can play a role in how mitochondria function, and also mitochondria themselves can store calcium. So these are relevant sections within the textbook. Okay, so mitochondria, they've been roughly known for about 170 years. They were first discovered in the muscle tissue of insects in the mid-1800s. And since then, we now know that it's an organelle that's found in most eukaryotic cells, including animals, plants, and fungi. And we've all kind of had heard this term, the powerhouse of the cell. So it's a very common phrase, and actually it turns out it was coined in 1957 in a Scientific American article. So we're all quite familiar with about mitochondria as powerhouses, but they do much more than that. Beyond making cellular energy, certainly one of the biggest functions of mitochondria, but they're involved in many other processes, including cell signaling, cell differentiation. They actually play a very active role in promoting programmed cell death or apoptosis, which I'll just very briefly touch upon, hopefully today. But towards the end of my lectures, I'll get into great detail about how that works. And they also play either a direct or indirect role in controlling the cell cycle and cell growth. All right, so we've all kind of familiar with this structure. It's a double membrane structure. It comes in various sizes depending on the cell and the state of the cell, but generally it's got about 1 to 8 micron length and roughly 0.1 to 0.5 micron diameter. It's got an outer mitochondrial membrane, it's got an inner mitochondrial membrane, and in between is the intermitochondrial membrane space. Intermembrane space, I should say. And then in the very center of that is what we call the mitochondrial matrix. So we'll talk in a little bit more detail, but there's a lot of proteins found in both membranes that have certain functions. And in the matrix, we find enzymes involved in the Krebs cycle, tricarboxylic acid cycle, citric acid cycle. It's all the same thing. We've all learned that. We all hate it. But there's also DNA, which we'll talk about in a little bit, and ribosomes and transfer RNAs. So the mitochondria is almost like its own little entity itself, like a little beast living within. And we'll talk about that. That's actually not far off from the truth. So a little bit more focus on some of the proteins that are found in the membranes. These membranes play really important roles in the function of mitochondria. At the outer mitochondrial membrane, we have proteins called porins. They're basically little channels that let stuff in and out. It could be ions, it could be small molecules, metabolites. And so a major one is called the voltage-dependent anion channel, or VDAC. So that's a major one that regulates the flow of material, as I say, metabolites, ions, other things in and out from the cytosol into the intermembrane space, and vice versa. It can go both ways. There's other forms as well. And then the inner mitochondrial membrane also has some proteins involved in transport, import ions, metabolites, other things. But more importantly, it has these vast, very big complexes of the electron transport chain. So we'll talk in a small amount of detail about complex 1, 2, 3, 4. I'm not going to reiterate all the metabolism and biochemistry. You've had that beaten to death, and people find it incredibly boring. So I won't get into that in great detail. But what's important to know is that these electron transport chain complexes ultimately are used to reduce oxygen, and they also, as we'll talk about in the next slide or so, cause an electrochemical gradient that's used by ATP synthase to make ATP. So this process is called oxidative phosphorylation. People have kind of heard that between high school and first year biology. So it's also important to note that, when we look at the membranes within the cell, particularly the inner mitochondrial membrane, has about the highest protein content. So these proteins are either embedded directly in or associated with other proteins that are embedded in the membrane. So proteins that are embedded in the membrane we call integral membrane proteins. And so we have a lot of these proteins in the inner mitochondrial membrane. It's one of the most proteinaceous membranes in our bodies or cells. Okay, so we're all sort of familiar with biochemistry, so I won't beat it to death too much, but just to refresh your memory, that in the cytosol we have glycolysis, and it goes through, glucose is broken down through a whole bunch of steps with a whole bunch of enzymes that nobody cares about, and ultimately will generate pyruvate. And so the pyruvate can be imported into intramembrane space, and there's a transporter that will transport it into the matrix. And then there's a number of enzymes that will work on it, and I won't get into all the enzymes because this is not biochemistry. It will be converted to acetyl-CoA, and that's kind of jumpstarts the process for it to be further broken down in the citric acid cycle or the Krebs cycle, or whatever cycle you like to call it. And the important part of this is it's going to generate NADH, nicotinamide adenine dinucleotide. The H is the reduced form. The plus is the oxidized form. And so you make a lot of reduced NADH. And then that is important because electrons from NADH can be used to donate into the electron transport complexes. So we talked about complex 1, complex, well, 2 is not shown here, 3, and, oh no, 2 is there. I don't know why they put it there. Anyways, electron transport complexes. Ultimately, the electrons are moved to completely reduce oxygen to water. And at the same time, there's the concurrent transport of protons. So you see that H there, the proton is transported across the inner mitochondrial membrane, and it will accumulate in the inner membrane space. So you actually cause a slight lowering of the pH in that space because you have the accumulation of these protons. So that creates that proton gradient. And then that can, because it's you have this buildup of protons, they can be moved back into the matrix via ATPase synthase, which will convert ADP to ATP, and that can be transported out and then enter into the cytosol and fuel all sorts of important processes. Right? So we've all kind of covered this, I think, to various extents. I'm not going to beat it to death. I'm just going to put it here as a reminder. The textbook has a bunch of other stuff that I don't really care about. It's not important. So just understand the idea of, okay, how does it make energy? We're all kind of familiar with oxidative phosphorylation. The idea that the Krebs cycle makes NADH, and that's going to help donate electrons to the electron transport chain, and also protons, which can move from the matrix into the intermembrane space. And then that can help create a gradient, and then which can be exploited via ATP synthase to help make ATP. And it makes it quite well. We get a lot of ATP produced, anywhere from 34 to 36 ATP. So that's great. Okay, this is where it gets interesting. So a little bit of a history lesson. This dude here is Otto Warburg. He got a Nobel Prize in the 1930s for discovering the respiratory enzymes in mitochondria. So that was a really important thing. But along the way, he was some of the early cell biologists being able to culture certain types of cells. I can't remember, don't quote me on it. It might have been frog eggs or sea urchins or something, I don't remember. He started with that, but then he moved on looking at normal cells and cancer cells in culture. And so as I just talked about, most cells are going to take glucose, they're going to take it up, and they're going to break it down first in the cytosol by glycolysis, and then it's going to generate stuff. It goes into mitochondria, Krebs cycle, ATP. We just already covered that. So this depends on oxygen. So oxygen is really quite important to this process. And although there's a lot of steps, the payoff is you make a lot of ATP. When we look at cancer or tumor cells, it's a little different. So when tumors are growing, cancer cells grow, and they start to form a ball known as a tumor. They grow quite fast. That's a feature of cancer cells. And what happens is the cells in the middle don't have enough oxygen. And if they don't have enough oxygen, they'll use anaerobic glycolysis, and they'll generate lactate. So it does it fairly efficiently, but it only makes 2 ATP. Depending on how you do your math, some people say 4, but I say 2. So it doesn't generate a lot of ATP, but it's able to do it in the absence of oxygen. And to compensate for the weak payout, cancer cells upregulate glucose transport. They sock up glucose like nobody's business. That's part of why you're looking, like if you go, you have cancer, and you see an oncologist, quite frequently what they'll do is they'll give you radio-labeled glucose. And they put you in a positron emission tomography or PET scanner. And where the tumors are will all light up because they're sopping up so much glucose. So the cell, this gets them by and they continue to grow, but then they send out signals to cause blood vessels to form into the tumor. It's called angiogenesis. And so that allows new blood vessels to infiltrate into the tumor. Now lots of fresh oxygenated blood come in, they have lots of oxygen. But what's interesting is that the cancer cells still rely on glycolysis for their energy needs, which seem to be quite paradoxical because it doesn't make a lot of ATP. So this is what we refer to as aerobic glycolysis, is that the breakdown of glucose to lactate occurs primarily in these cancer cells, even when oxygen is present. So the question is, why would they do that? Well, first off, it reduces the amount of mitochondrial activity because they need oxygen. Even though oxygen is present, they elicit little breaks on mitochondria to prevent them from being very active. So why would they do that? It turns out that by having high levels of glycolysis, there's a whole bunch of really complex glycolytic intermediates So the upper part of glycolysis, which I'm not going to get into because it's got a billion different enzymes that nobody wants to talk about, but the important thing is that that upper part of glycolysis, it gets shunted away into these little side hustle businesses in terms of making molecules that can result in the formation of nucleotides to help make DNA, or make more amino acids or lipids. It's called anabolic metabolism. It's a way for a cell to bulk up. It makes more macromolecules to help make a more cell. So if a cell's going to divide and it's this big and it's only got this much, if it divides in half without bulking up first, it'll make little tiny wimpy cells. So it kind of bulks up. It's like pumping iron, so to speak. It's building all the precursors to make a new cell. So that anabolic metabolism is what allows cancer cells to proliferate so readily and make more cells, because they're making all the building blocks of cells. Now there's another aspect, which I won't get into too much detail, is that when mitochondria are less active, they're less likely to leak out stuff that can trigger programmed cell death or apoptosis. So that's another feature of cancer cells, is they adapt mechanisms to resist apoptosis. Our immune cells will quite frequently attack cancer cells and deliver signals to trigger apoptosis, in many cases through the mitochondria. So by having mitochondria kind of quietened down, they're less likely to initiate apoptosis. So the aerobic glycolysis, named and more commonly known as the Warburg effect, named after Otto, it does two things. It allows the buildup of intermediates to help make more cells, i.e. nucleic acids, amino acids for protein, lipids for membranes, and it also helps quieten down mitochondria to prevent apoptosis from occurring. So what's interesting is Otto Warburg was quite convinced that this was going to be the Achilles heel for cancer, and this could be a very effective, by interfering with this somehow, because cancer cells, at least back then, we thought were uniquely eliciting this metabolism. What happened in the 1950s is that we discovered DNA and the structure of DNA and the genetic sequence, and then discovered that the mutations were driving most cancers. And that poor Otto was kind of ridiculed for because he discovered this phenomenon in the 1920s, so 30 years later he was still beating that aerobic glycolysis drum and nobody was listening. It turned out about 20 years ago, 18 years ago, it was rediscovered, and it turns out he was right all along, is that it actually is a very important metabolism for cancer cells. It's a lot more complex than what I'm just saying here, but many drug companies are trying to manipulate this as a weakness of cancer cells that they can exploit with chemotherapeutic agents. Okay, and in the textbook, again, I thought, well, I better put some textbook material on this. They actually do have one slide or one picture on it. It's just reiterating what I was saying. In differentiated non-transformed cells, if oxygen is present, they're going to use oxidative phosphorylation, makes lots of ATP. In a circumstance where there's oxygen deprivation, it can happen sometimes. So, for example, if you have a stroke and a blood vessel is blocked, there's inadequate oxygen, and by default, the cells will rely on glycolysis and make lactate. In tumor cells, as I mentioned, they'll utilize this type of metabolism, again, because it helps for anabolic metabolism and an anti-apoptotic mechanism. We now know that there's other cells, non-transformed cells, particularly stem cells, interestingly, that also use this type of metabolism. So it's not just unique to cancer cells. We know that highly proliferative cells, and including stem cells, also employ this metabolism, and it actually is important for self-renewal and maintenance of stem cell populations. That's something that I'm also examining in my lab in terms of neural stem cells in the brain, seem to rely on this metabolism also. So that's one of the things that we need to take into consideration if we're designing drugs to mess up this metabolism. It might work really well in cancer cells, but it could have some bystander effects on healthy cells as well, particularly stem cells. Okay, so I mentioned a little bit about DNA and mitochondrial DNA. So it's a circular type of DNA that's found in the matrix of mitochondria. Where did it come from? Well, it turns out that you can go back about 1 billion plus years ago, when we now had photosynthetic organisms, usually this algae, but then moved on to plants, started producing oxygen. And so there were bacterial cells that were actually utilizing that oxygen to make energy in a manner very similar to how mitochondria do it. So the theory is that we had a sort of an ancestral cell. It was a eukaryotic cell, it had a nucleus, and probably had some other organelles. And it was probably using mostly glycolysis to make energy. And at some point, this cell engulfed a bacterium. And this bacterium eventually conferred an advantage. So it's called an endosymbiotic relationship. So it's like we kind of work together and that we both benefit. You scratch my back, I scratch your back. And so by the bacteria becoming engulfed in the cell, it had some benefits in terms of now the cell could kind of take care of it a little bit, but it would feed back in the form of making more energy, ATP. So in this case, there was, a bacterium that had circular DNA, it had an outer membrane, and it also had ATP synthase. And so it was using a kind of a rudimentary type of oxidative phosphorylation process, and that got basically incorporated when it over time, many generations, over millions and millions of years, is that it formed its own entity known as what we know now as a mitochondria. And so the mitochondria have retained some of the DNA, ancestral DNA from the bacterium. They've also retained some of the pieces that are important, like ATP synthase and a few electron transport chain complex members. But what happened is some of this DNA ended up going into the nucleus. So when we talked about all those proteins that really play really important roles in mitochondrial membranes, most of them are encoded by nuclear genes. But there's some that are encoded by the mitochondrial genome. And so there's been some theories and reasonably good evidence to assess why this would happen. It turns out that some of the genes that are the proteins encoded by those mitochondrial genes make proteins or polypeptides that are kind of big or very hydrophobic and very difficult to get it into the mitochondria. So you make it at the source, and then it can be incorporated into either the inner or outer membrane. It also turns out that there's a DNA that encodes for transfer RNAs. And some of those are very large and difficult, again, to import. So make it at the source. And which I don't know if we'll have time, probably not, but we'll talk a little bit about the latest discovery, i.e. last month, discovering that activation of mitochondrial DNA plays a really important role in memory. And so in terms of neurons in your brain. And it can do it very quickly. So one of the advantages of having mitochondrial DNA is it's much more flexible. You can turn it on and off very quickly in response to changing conditions. As opposed to the nucleus, maybe you'll have a whole series of events to lead to activation of transcription factors. Maybe those transcription factors have to go from the cytosol into the nucleus. We'll talk about that much later in the course. And there's many steps involved in genomic DNA transcription. There's fewer steps, and it's faster in terms of mitochondrial DNA. Okay. So again, some other interesting tidbits about mitochondrial DNA. So depending on the cell type, there's actually a great variance. Some cells that require a lot of energy tend to have much higher numbers of mitochondria. Cells that require less energy have lower numbers, but typically if we average it, there's about 1,000 to 2,000 mitochondrial DNA molecules in each cell. There's some exceptions. So if we look at a human egg, it has 500,000 mitochondrial DNA molecules, whereas a sperm only has 100. So this is what leads to maternal inheritance of mitochondrial DNA. It's almost exclusively from the mother. There's very little to none that comes from the father, which is kind of interesting. The other thing to take into consideration is what happens when cells divide. And so when cells divide, they want to try to ideally equally portion or segregate an equal number of mitochondria into the daughter cells. And for the most part that happens, but there are some cases where it doesn't happen, which can be problematic. So this brings up the concept of mitochondrial heteroplasmy. So mitochondrial DNA, just like nuclear DNA, is susceptible to damage, DNA damage, which can introduce mutations. So it doesn't mean that every single mitochondrial DNA has mutations, but quite frequently is you get a subset of mitochondrial DNA within a mitochondrion that has mutations. So, and those mutations can be passed on in terms of when a cell replicates. Now, when a cell replicates, and we'll talk about in a little bit, is that quite frequently mitochondria will undergo fission, they break apart. That's not always, but quite typically what happens when cells are dividing. They actually split the mitochondria, and we'll talk a little bit about fission in a minute. And so what happens is when cells are replicating and they split mitochondria and then half goes to that daughter cell, half goes to that, you can get a certain level of mitochondrial DNA that has mutations. So if we look at the originating cell here, it's occurred, so the green mitochondrial DNA is happy DNA, there's no mutations, it's what we call wild type, and the purple has incurred some kind of mutation. It's usually deleterious, and it's going to affect some of those critical mitochondrial genes they need for particularly electron transport chain activity or translational abilities. So what happens is when cells divide, you can end up with a situation where maybe one cell basically gets pretty well all healthy wild-type DNA, whereas another cell may get a mixture. Or you may get a cell that has almost exclusively mutated DNA, mitochondrial DNA. So when you have the mixture situation, that's called heteroplasmy or heteroplasmic variant, and you can have homoplasmic all wild type or homoplasmic all mutant. To be honest, it's not quite like that. In reality, you get a mixture. So this is kind of going backwards compared to here, but we have, let's say, a wild type cell that has all wild type healthy non-mutated DNA. That's in the homoplasmic wild type state. And you may end up with some cells that end up with a few mitochondrial mutations, which can then get segregated. And as they get segregated, they can either get diluted or accrue more mutations. And so when you have a mixture of both wild type and mutant DNA, as I referred to, that's the heteroplasmic state. But there's a sort of a critical threshold where you accrue more mutant than wild type. And then usually when you get to the state, particularly if you're in a homoplasmic mutant state, that's when disease will manifest. So when you reach that critical threshold where you have more mutant mitochondrial DNA than wild type, it generally leads to compromised mitochondrial function. If it's just chock full of mutant DNA, those mitochondria are not going to function properly, and that's going to cause a whole bunch of problems. So the more that you have of the mutant mitochondrial DNA, it can lead to more severe diseases. What are kind of inherited mitochondrial diseases? There are almost thousands of them. So I don't have time to get into it. I just picked a couple. There's various diseases, neuromuscular diseases, neurodegenerative diseases, diseases of the eye, the optic nerve, all sorts of things. So these are, and quite frequently, it affects most adversely cells that rely on a lot of mitochondria. So it could be muscle cells or neurons. They both rely on, because they have incredibly high energy needs, if those mitochondria aren't functioning properly, you're going to have adverse conditions, a bad situation for that particular tissue, and that can lead to disease. Another thing that is interesting is that, not necessarily leading to disease, but as we get older, we accrue more mitochondrial DNA mutations. And there's evidence that this accumulation of mutation over time may actually contribute to the aging process of the cell. Our cells are just simply not able to make ATP as easily. So it's almost like a metabolic insufficiency as we get older. So there's lots of evidence that certain tissues in our body just accrue a ton of mitochondrial DNA mutations, and the mitochondria are just not keeping up with the needs of the cells. And that is one of many things that contribute to aging. So another interesting phenomena, actually is fairly recent, discovered about 20 years ago, are these things called tunneling nanotubes. They're kind of interesting, and I've seen it in culture too. They're kind of freaky. So you'll see cells in culture, and sometimes you see the little stringy, but sometimes it's very difficult to see, this tiny little tube that connects one cell to another cell. And this is what we call a tunneling nanotube. So they directly, it's an extension of the plasma membrane. So this tube is an extension of the plasma membrane that connects the two cells together and allows transference of cytosolic content from one cell to another. So they're fairly thin, so 5 to 300 nanometers or 0.3 microns. Their length can vary quite a bit, from a very short distance to 200 microns, which is actually quite a bit, longer than several cell diameters. And not only can they transfer molecules, they can also transfer organelles, including mitochondria. Mitochondria. So this is what we refer to as intercellular mitochondrial transfer. It turns out, again, this is emerging evidence. It's not fully understood, but they're finding more and more studies are finding that this nanotube can have both good and bad roles. So in some cases, it can actually help a cell that's struggling. So there may be a cell that has insufficient number of mitochondria and it's not meeting its energy needs. And buddy next to him has got quite a bit of mitochondria, and it shoots out a tube and says, here, I'm going to help you out. It transfers a few mitochondria, or maybe a bunch, over into that cell. It can help restore the function or the metabolic state of that cell. And this doesn't happen just between two cells that are of the same type. You can have different cell types that connect via these tubes. One example is found in the brain, where astrocytes and neurons can form these tubes. And sometimes astrocytes can help neurons out by giving them some extra mitochondria, or vice versa. But tends to be astrocytes don't always need mitochondria as much. They have a different, astrocytes tend to use more aerobic glycolysis. That's another cell type. And therefore don't need as many mitochondria. And so they can kind of donate to neurons to help them out when they're struggling. So that's one aspect of how they can help modulate and help out cellular metabolism. But in some, and then again, that's part of mitochondrial quality control. So how this happens and the mechanisms is still not entirely certain, but there may be signals that a struggling cell sends out to its neighbors, I need help. And so this can cause the formation of these nanotubes where a healthy cell can now donate to a cell that's got struggling mitochondria that are not functioning. So again, it's part of mitochondrial quality control, helping out and replenishing mitochondria, healthy mitochondria in a cell that has a lot of dysfunctional mitochondria. And not to get into too much, again, because to be honest, I'm not that familiar with the literature, but there's lots of evidence of cancer cells communicating between themselves and also healthy cells in terms of exchanging mitochondria. And this can manipulate the metabolic state of cancer cells and their adjacent cells. There's cancer niches as well. There's something called cancer-associated fibroblasts. These are non-transformed cells, and the cancer cells actually starts to influence these adjacent fibroblasts and start to mess them up and exploit them and manipulate them to help the cancer cells. So even though the cancer cells are not transformed, they form a niche almost themselves and can actually have this metabolic shuffling, part of which is mediated by these nanotubes. And there's also evidence that immune cells exploit this as well to help meet their metabolic needs when there's maybe infection or inflammation. Okay. So I mentioned that mitochondria are dynamic, that they're not just this little round pill-shaped oval thing. They can be, but if you actually look at cells in a dish, and sometimes in tissues, and you stain, so this is a HeLa cell, and that little dotted line represents the nucleus, and all these little, mostly green squiggly things are actually mitochondria. So they're not little ovals, that they form these long tubular structures that sometimes branch. And so some of this is related to how mitochondria either stick together, called fusion, or break apart, called fission. And the textbook is showing, I guess these are some elongated mitochondria labeled with some fluorescent marker, and over time you can see that these two start to stick together. That's called fusion, and this one we can actually see it breaking apart, fission. So you can monitor this in actual live cells, depending on how you label them with fluorescent markers, which Dr. Pemberton will talk about. So it turns out that cells that have more of these elongated, tubular, branched mitochondria tend to respire more efficiently, are more metabolically active, and produce more ATP. Why is that? Well, it's about basically increasing the surface area. You have more membrane, you have more surface area, more ability to interact with oxygen, and more electron transport chain complexes that can process NADH from the Krebs cycle and using oxygen to elicit ATP production through oxidative phosphorylation. So healthy unstressed cells tend to have these complex, extensive networks of fused mitochondria, and they make high ATP. Stress cells tend to undergo more fission, and they have more fragmented mitochondria, so they have lower ATP production. So the sort of the state of the cell can be influenced. So if the cell is stressed, it tends to undergo more fission. If it's healthy and happy, it tends to promote more fusion, and that can help increase oxidative phosphorylation. You make longer mitochondria. Oh, there's one thing that I forgot right at the beginning. I can't believe I forgot this part. Is that the undulating part of the inner membrane are called cristae. So you see those invaginations of the inner membrane. So you have more of those, you're creating more membrane, more surface area. So those branch mitochondria, elongated mitochondria, have more cristae. So so you have more membrane that's folding in and out, forming those almost like finger-like projections. Again, increases more electron transport chain activity. More membrane, more proteins, more surface area, more ability to interact with oxygen, more ATP. Okay. So what regulates this? So so membranes just don't stick together. It's energetically extremely unfavorable. So you have to get over this energetic hurdle. So you need proteins to help out with it. And you need energy. So there's a number of GTP hydrolyzing enzymes. So GTP can be broken down to GDP. Well, this is a common theme in cell biology. We'll talk about it many times. And it's been frequently used to generate energy to overcome energetic barriers like membrane fusion. And so these GTP hydrolyzing enzymes belong to the dynamin family. They play a major role in membrane fusion. We'll talk about that in a little more detail when we talk about receptor-mediated endocytosis. We're not there yet. But there's three proteins we'll talk about that participate in fusion. The first is mitofusion 1 and 2, or MFN1 and MF2. They're integral membrane proteins, so the part that traverses the outer mitochondrial membrane. They have another part that sticks out into the cytosol. And so what can happen is they receive signals, which I'm not going to get involved in, this is complex, that promote their interaction. And so they butt up against each other, get very, very close, and as a result, it helps bring the membranes in close proximity, and then they fuse. So mitofussion 1 and 2, found at the outer mitochondrial membrane, get close together to allow the fusion of these two outer mitochondrial membranes. Two mitochondria now fuse together. And then, once they fuse together, another protein called APA1, which is found, another integral membrane protein found in the inner mitochondrial membrane. These two meet, and they get close together, and they permit the fusion of the inner mitochondrial membrane, and now we have two mitochondria now become one. The opposite is fission, and the major protein that's involved in fission is called DRP1. Again, it's part of the dynamin GTP hydrolyzing enzyme family. But what it does is it, through various signals, which I'm not going to get into, will bind to the outer chondral membrane, and they stick together. It's called polymerized. Like a whole bunch of those DRP proteins all glom together. This starts to promote a pinching of the membrane, and it continually basically constricts the membrane so much so that the membrane will now split apart. And so, again, GTP hydrolysis is required for all these events because it's energetically very demanding. And so the major part is to understanding what are the players involved in fusion, sticking two mitochondria together, and what are the proteins involved in splitting apart. The main one, there's many, I'm just picking the most prominent one, is DRP1 is involved in fission. It is not an integral membrane protein that floats around. It's a soluble cytoplasmic protein. It receives certain signals to bind to the outer mitochondrial membrane and then associate as a polymer. Many of them stick together and form the pinching, which leads to the breaking apart of the mitochondria. Okay, so we've only got 1 minute, so I think I'll probably pause there. But just to announce that all content up to this lecture slide is testable on Sunday, and we'll post more information about the test on the weekend. Yeah, just give me a minute just to disconnect here. Just because I know there's another one.
Okay.